Influenza, usually just called the flu, may be the most famous virus in the world. But how does this tiny cluster of protein and RNA lead to the deaths of roughly 500,000 people every year? Here's what you need to know. Influenza has a genome of -sense single-stranded RNA broken into 8 segments. Its capsid is also enveloped in a lipid bilayer that it steals from its last host. This lipid bilayer is full of hemagglutinin and neuraminidase, 2 proteins that help it bind to cells and later get released from them. It also has an M2 protein channel which pumps ions, but more on that later. Overall, the -sense RNA genome classifies influenza as a class 5 virus, meaning it also must have its own RNA polymerase. Influenza is spread via the microdroplets formed when you sneeze or cough. Those droplets can hang around for a long time in the right environment until they get inhaled by the next host. Once they enter the host, they work their way down into both the upper and lower airways. Their target? The epithelial cells of the respiratory tract. When they reach those cells, influenza virions have a few hurdles to jump. The first is the mucus layer covering those epithelial cells. Along with the host immune cells patrolling, cilia on the cells move the mucus, and the virions along with it, out to the gastrointestinal tract, where they would be destroyed. Many are beaten by these forces, but all it takes is a few virions to make it through for the infection to begin. Even if they make it past the mucus layer, virions are still not infectious until their hemagglutinin proteins are cleaved. Fortunately, specialized cells in the respiratory tract called club cells, formerly Clara cells, produce the enzyme tryptase, which does exactly that. The need for these cells is what isolates influenza from spreading out of the respiratory tract and to the rest of your organs. Once hemagglutinin is cut, the virion binds to sialic acid located on the surface of the epithelial cells. This binding activates receptor-mediated endocytosis, a process of enclosing the virion in a portion of the plasma membrane, creating an endosome. Now, the goal of the virion is to reach the nucleus of the cell so that it can replicate its genome inside. This means it must somehow escape the endosome. Since the membrane it is bound in comes from the membrane of the cell itself, it contains the proton pumps that regulate charges in and out of the cell. These proteins are activated to fill the endosome with protons. Usually, this would signal the end of anything inside the structure. But influenza imports those protons inside its own envelope using that M2 protein we mentioned earlier. This acidification activates the binding of influenza's envelope with the membrane of the endosome, allowing the genome and various proteins of the virus to be released and work their way into the nucleus using nuclear localization signals present on the viral proteins that remain bound to the segments of RNA. Once the genome is inside the nucleus, replication can begin. Immediately, the RNA polymerase that came with the virus begins to create complementary positive messenger RNAs. These RNAs then steal the 5' caps off of the host's own RNAs, so that when they reach a ribosome, it will recognize them as one of its own and create the needed protein. This also serves the dual function of stopping host mRNA from competing for ribosomes. These proteins are created in the endoplasmic reticulum, and the majority move to the Golgi apparatus and use it to facilitate their route to the membrane. 2 of the proteins made are NIP and M1, which will be important in a moment. The genome itself also gets replicated with polymerases to create positive sense complementary RNAs. These are used to produce lots of new -sense genomes for the virions to come. M1 and NIP return back into the nucleus to bind to the new viral genomes. NIP specifically facilitates the export of the genome out of the nucleus and into the cytosol. From there, the virion can begin the journey back out of the cell. Those proteins that rode the Golgi out to the edge of the cell incorporate into the membrane and create islands of viral envelope proteins. The neuraminidase clears out the sialic acid in the area so that when the virion is ready to bud, it won't accidentally bind to the same cell it just left from. Viral genomes that have left the nucleus hitch a ride on the endocytic pathway, which moves endosomes along microtubules out to the cell's membrane. Once there, they associate with those protein islands in clusters of 8 segments and bud off, ready to infect another cell. And that's what you need to know.